All right. So welcome back. So we have the second semester of lectures. Today's speakers is Rikhia Ghosh. So Rikhia is, well, originally from India. So she has been graduated from the Indian Institute of Science in 2015. And then she has quite a professional career in different institutes in Germany, from 2016 to 2022. She was a postdoctoral fellow at the Max Planck Institute of Colloids and Interfaces in the group of Reinhard Lipovsky. From basically last year till now, she's a postdoctoral fellow at the Icahn School of Medicine at Mount Sinai. So her interests are broad, basically from development of machine learning-based approaches for the understanding of GPCR, as well as the development of coarse-grained models for biological membranes. I guess that's a little bit of the topic that she will bring to us today. And well, she has a long expertise also in conformational analysis. So today's talk is going to be understanding the molecular origin of emergent behavior in membrane remodeling. So Rikhia, the floor is yours. We are happy to have you.
Thank you, Dr. Poma. It's been a pleasure to be able to present my work here. So Dr. Poma already gave a very nice introduction about my background. I would still give you an overview about my career trajectory. So I hail from India. Actually, I was born and raised in a city called Calcutta. That's how it's known in Western countries. So in our language, it's called Kolkata, where I did my bachelor's and master's in chemistry, and my specialization in master's was in physical chemistry. Followed by that, I decided to do my PhD from an institute in India, which is located at the southern part, which is, and the institute is known as Indian Institute of Science. So in my PhD, I started working on classical physical chemistry problems like binary mixture and understanding water dynamics, water structure and dynamics. But I slowly gravitated towards more problems in biophysics, because that's something I was always very intrigued about. And I started decoding and understanding protein hydration dynamics and protein folding dynamics, how the conformation of protein changes in different aqueous solvents. So that's where my interest in biophysics started developing. After my PhD in India, I moved to Germany in a small city called Potsdam, which is in the suburb of Berlin, at the Max Planck Institute of Colloids and Interfaces. So here I took a leap and I went from physical chemistry to condensed matter physics, but the application was always in biophysical problems. So here I worked on primarily on membrane remodeling, and that's the work I'm going to present today. Currently, I am working at in US, in New York, in Mount Sinai Hospital. So here again, I took a leap, and I am working on more, I would say, new age problems like drug discovery, like under the broad domain of drug discovery. So that's something that's very interesting and challenging at the same time. So in terms of work, this is a brief sketch of what I did during different times. So in Indian Institute of Science, or the acronym is IISc, I worked on binary mixtures, as I said. Then I worked a couple of problems which involved polymer conformational behavior and how polymer conformation changes in different kinds of binary mixture. I also worked on protein folding dynamics, and basically I tried to estimate the free energy of protein unfolding for different protein families. At Max Planck Institute, which is I'm going to present today, I primarily worked on membrane morphological behavior. So it was, I mean, the first I started understanding how membrane remodeling works and what is the role of the membrane mechanics there. And then I slowly took it forward to model different membrane remodeling behaviors in biomimetic membranes. And one of our very successful projects was understanding membrane endocytosis. And currently I work on GPCR signaling kinetics and AI-assisted drug design. So to give you a flavor of what is my interest, I work on understanding self-assembly process in biology. So what is very well known is that non-living molecules like proteins, lipids, DNA inside cells, they undergo spontaneous self-assembly processes through a cascade of ordering events to create cell, which build up further to create tissues and organs. And what is very interesting here is that these non-living molecules, small molecules like proteins and DNAs and lipids, they not only create cells, but they also create these extremely sophisticated nanomachines. Examples are um, the machineries which execute membrane scission or ion channels. And these concertedly work together to perform cellular functions. And one thing very important here is that the assembly of these molecules are as important as disassembly, because when that doesn't happen, that leads to different kinds of diseased states. For example, in case of amyloids, this leads to formation of amyloid aggregates if these assemblies don't spontaneously disassemble. Now, I come from this school of thought, or there is a big school of physicists who believe that in order to understand these processes which occur at intermediate scales, it's not necessary to know the movement of every water molecule or every atom in space, but one can have a rough idea of the main features of these molecules, like size and long range interactions. And based on these ideas, one can build coarse grain model of these nanomachines, for example. And because in that case, one can reduce the dimensionality of the degrees of freedom, and that would lead to the correct representation of the dynamics and the structure when these molecules assemble. So one can build a very nice model of the assembly and try to understand the mechanism of the assembly without really making it computationally very expensive. So that's the school of thought, and that's like my personal goal moving forward. That's where I'm aiming at, is to understand collective behavior of living system using and developing molecular and mesoscale models. So there are different channels of subjects which are very helpful to build these models. And these, like, one uses ideas from physical chemistry, soft matter physics. Of course, computer simulation is an integral part, and ideas of life science to build these models. And that's what I have been doing since I got gravitated towards understanding biophysical problems. So how does this whole pathway work? So in order to build this connection between the what happens at cellular level and how we can understand that at the level of by using computer simulation, one needs to understand first how the cellular machinery work, at least to have a brief idea. So here, the example I'm giving you is like the formation of different kinds of shapes in membrane. For example, the tubes or the pearl tubules. So this is an observation that in presence of condensed or dilute phases of liquid of protein assemblies, the membrane undergo shape transformation to form different kinds of tubules. Based on these observations, one can build up a hypothesis of the molecular mechanism. Based on this hypothesis, there comes the action of the computer models. One can build a toolbox by creating the building blocks in the coarse grain space. So, for example, since the membrane is involved here, one can build a rough coarse-grained representation of the lipids, which can look like a combination of beads which are bonded together, or they're with a specific value of angle, for example. And using these building blocks, one can create the structure. For example, here, in case of a membrane, one would like to build a membrane patch or a vesicle. And then one adds dynamics on top of that using different kinds of force fields, which would help us to further identify and determine the proof of concept, like how the self-organization of the proteins lead to the changes in the shape of the membranes. Now, with that, I would like to delve into what I did when I was at Max Planck Institute, which was primarily, the idea stemmed from primarily the fact that we wanted to understand the origin of membrane remodeling in a from a very simple perspective, from a physicist perspective. And the motivation, a short motivation of that is that inside cells, there are many organelles which have a membrane as an envelope. And during cellular trafficking processes, these membranes undergo spontaneous remodeling to form buds, and which carry cargo from one organelle to the other, and there are several processes involved which help carry out the processes. So, for example, a membrane buds out from the site of origin, and then it gets to the target site. For example, it goes from endoplasmic reticulum to Golgi complex. And so our goal here was to understand how this membrane remodeling happens. Like, it's a spontaneous active process, and we wanted to understand what's the mechanical origin of this membrane remodeling. I would just like to tell you that please feel free to stop me and ask questions. It would be nice to keep the session interactive. And please let me know if I'm going too fast. I will try to slow down. Yes. So here are different examples of membrane remodeling that I could collect from the literature. So this is very well known that membrane remodeling is an integral part of any active cellular processes, and different groups at different point of time tried to model the membrane remodeling in vitro as well as in computer simulation. So the first example is how membrane remodeling happens during mechanoadaptation. Then there is this example of membrane remodeling during cell division, which is also an in vitro study. This occurs using membrane-bound proteins. The third example is the one I already showed you, that membrane bending and tubule formation by low complexity domain of Fusin sarcoma, which occurs by liquid-liquid phase separation of intrinsically disordered proteins. And the fourth example is a computer simulation study of archaeal cell division by escort-3 filaments. So a generic question, like a common question for all these cases, is what are the role of intrinsic physical forces in driving membrane remodeling? So in order to understand that, one has to decouple the different phenomena that occur simultaneously and understand membrane remodeling at a molecular level. So if you look at a cell and if you zoom into the bilayer of the membrane, you will see that it's a bilayer of membrane composed of lipid molecules. And in order to maintain an equilibrium, the bilayer the membrane bilayer tries to keep the minimal membrane tension so that there is no pore formation or rupture of the membrane, and which means the bilayer tension is very close to zero. However, what is like not so intuitive here is that even though the bilayer tension is zero, that does not necessarily mean that the inner and outer leaflet tension will be zero as well. So the inner and outer leaflet tension can be substantial and still cancel each other to give a total bilayer tension of zero. However, if that is the case, that is the inner and outer leaflet tension are non-zero, the membrane will try to minimize those tensions, and that can only happen when the membrane curves accordingly. And this would lead to the membrane remodeling process. And this is what we tried to understand, that what is the role of membrane leaflet tension in executing membrane remodeling. And it's very well known that membrane remodeling happens by molecules of increasing complexity, which can be through small molecules and large protein assemblies like escort or coat proteins, bar domain proteins. But here we wanted to understand it in a very simple manner, that is how the membrane remodeling happens when there is an imbalance of lipids in the two leaflets. And the second case was how the membrane remodeling happens in presence of small solute molecules that get preferentially adsorbed on the membrane surface. So this is very similar to what I described about my interest at the beginning, that we wanted to understand the membrane remodeling process, and for that we needed to minimize the level of details associated to the lipid membrane. So we used a coarse grain model of the membrane, and we built up the model in order to create these vesicles of small unilamellar vesicles. And throughout the study, I have always maintained the size of the membranes constant, like the vesicles constant, which is of the diameter of the order of 50 nanometers. The force field that we used is a very well-known force field for dealing with these mesoscale level problems, and it is called a dissipative particle dynamics. And to give you an estimate, the potential kind of shows you how our dissipative particle dynamics force compares with the Lennard-Jones. So it is a soft pairwise repulsive force. And there is a conservative, like this force comprises of these three different segments. But what is important here, or the that gives the character to the different kinds of coarse grain beads, is the conservative force, or in particular this constant value Fij. So the lower the Fij, the more attractive is the interaction between two beads, although in the potential you won't see any attractive well like in Lennard-Jones. But the force scales in such a manner that the higher the value of Fij, the stronger is the repulsion between two kinds of beads, and the lower is the force Fij value, the more attractive, so to say, is the interaction between two kinds of beads. And to give you an estimate, like this force field is one can create, for example, in case of water molecules, three water molecules together forms one coarse grain water bead of the dissipative particle dynamics. And because we have these small unilamellar vesicles, these are composed of lipid molecules of these architecture, where there are three head groups and two tails, each having 6 beads, and they are bonded using a bonded interaction value, bonded, and there is also angle constraints given to them. So the first idea that I would, the first work that I did and I would like to talk about is the understanding membrane remodeling by transbilayer asymmetry. And the motivation came from the fact that there are several studies, but there is this one study I particularly picked, which is very recent, is that membranes in cells are typically asymmetric in nature. And by asymmetric, I mean not just in terms of the types of lipids, but also the number of lipids in the two leaflets. So by asymmetric, I mean the two leaflets do not have the same number of lipids, but they are very inhomogeneously distributed. Because we wanted to keep it simple, and we wanted to understand the effect of the lipids distribution when it is inhomogeneous. So we had only one type of lipid, but we carried out this study by distributing the number of lipids in an inhomogeneous manner in the two leaflets. And in that case, we wanted to understand how the mechanical properties of each bilayer and leaflet changes, and how that plays a role in membrane remodeling. So to make it more comprehensive, I have created this schematic diagram. So let's say we start from a vesicle with a specific number of lipids in the two leaflets, and then in a systematic manner we added lipids in the outer leaflet and subtracted lipids from the inner leaflet, which would eventually create an inhomogeneous distribution. And this is how it looks like. So the first, like, we started from these many lipids in the first case, and then we in a very systematic manner we subtracted lipids from the inner leaflet, added lipids in the outer leaflet, and that in that way we created a few sets of vesicle. Now the first thing we wanted to understand here is, just by changing the number of lipids in the two leaflets, how that changes the mechanical properties of the vesicles. So one thing is very important here is to understand is that because we want to avoid pore formation or membrane rupture, we always made sure that the bilayer tension is maintained at 0 or very minimal, like close to 0. And we estimated that by calculating the total bilayer tension from plotting the radial stress function. So, and manually, how one can do that? One can actually uh, um, tune the number of water molecules. So these vesicles are not empty like how I show here. They are they're filled with water, but one can tune the water molecules inside, the number of water beads, let's say coarse grain beads, because that's what we are dealing here with. So one can tune the water beads inside and outside, and therefore can minimize the tension to a very low value. And that's what we did. And what we found is that as we increased the fraction of lipids in the outer leaflet, that led to tension imbalance in the two leaflets. So with increasing fraction of lipids in the outer leaflet, the tension in the inner leaflet went up, whereas the tension in the outer leaflet went down, as you can see in this plot. And that was already very interesting. And what was also interesting to find that by changing the number of lipids in the two leaflets, we could also arrive at a particular special case where the two leaflets have same tension, and there is 0 tension, which means this is a particular case where the two leaflets have 0 bilayer tension and 0 leaflet tension. So this was something we could estimate just by doing this systematic kind of study. So the first step was to understand that transbilayer asymmetry leads to finite leaflet tension. After that, what we did is we tried to understand if one such kind of transbilayer asymmetry can play a role in membrane remodeling. So we carried out the in order to execute that, we deflated the vesicles in an equivalent manner, like how biophysicists do that in experiments. That is, we deflated the vesicles by taking water out of interior of the vesicle. And we saw that while we did that, based on the fraction of lipids in the outer leaflet, the membrane underwent various morphological changes. So the first case where the outer leaflet tension was very low, which is like here, the membrane formed this kind of shapes, which is a precursor of membrane division process. That is, the membrane formed these outbuds. In the case where the vesicle tension, the outer leaflet tension was low, the membrane formed the other kind, like it reverted the morphological change, and it formed this kind of inward invagination and formation of inward buds. And what was highly interesting is that as we increased the fraction of lipids in the outer decreased the fraction of lipids in the outer leaflet, it just did not like it formed the inward bud, but at the same time this inward bud underwent a scission to form an a vesicle inside the mother membrane. And that was very cool observation. So basically what we did then, we varied the fraction of lipids in the outer leaflet, and also varied at the same time the vesicle volume. And just by playing around with these numbers, we could arrive a plethora of different shapes. So we, at low fraction of lipids in the outer leaflet, we could form these kinds of like inward shapes. And then we could form these oblate shapes, prolate shapes, and finally outbuds. So now we wanted to understand the mechanical origin of this process, so why this is happening. And in order to do that, we calculated the spontaneous curvature, which is an intrinsic measure of membrane curvature, and one can do that from the first moment of the stress profile. And what we saw is that with increasing fraction of lipids in the outer leaflet, the spontaneous curvature also scales linearly. So at low fraction of lipids in the outer leaflet, the spontaneous curvature is negative, whereas at higher fraction of lipids in the outer leaflet, the spontaneous curvature is positive. Now, what that means, what is the physical picture there? So this kind of made everything clear related to the observations. So when the outer leaflet tension is is negative or very low, that means that the outer leaflet is compressed, that there is an excess number of lipids in the outer leaflet compared to the inner leaflet, because at this level the fraction of lipids in the outer leaflet is high. And in this case, the outer leaflet, given a chance, would like to expand. And when we deflate the vesicle, this expansion process is possible, and therefore the outer leaflet remodulates itself to create this kind of outbud, because that creates a space in the outer leaflet, which increases the area per lipid and also minimizes the tension. Therefore, at higher fraction of lipid in the outer leaflet, the outbud forms. And the opposite case happens in case of low fraction of lipid in the outer leaflet. In that case, inner leaflet is compressed, and on deflating the vesicle, the inner leaflet would like to expand, and that's only possible when the inner leaflet forms this kind of inbuds. So that was very like this was a revealing picture for us. Even though it was always there, it was never really systematically understood. So therefore this study opened to a channel of further studies that we wanted to proceed with. So as the next step, we wanted to understand how can one process and understand membrane remodeling by solute adsorption. And this is more tangible for experimentalists, because this was always the case that the biophysicists working in our department would always talk to us about this, that they would always see if there is a sugar in a sugar solution or ionic solution, the membranes or the giant unilamellar vesicles, they often undergo morphological changes to form outbuds or inbuds or tubes, like different kinds of morphologies, but they never really could pinpoint what was happening there. So it was a fun study to understand what exactly happens when small molecules representative of sugars or ions, or one can even imagine intrinsically disordered proteins that preferentially absorb on the outer lipid on the outer surface of the membrane, can influence the shape transformation. So in order to do that, first we characterize the solute conformation, solute, how the solute solution is, like the aqueous solvent. And we wanted to understand that by first plotting the phase diagram of the solute. And we plotted the phase diagram as a function of solute mole fraction and solubility. And therefore, we characterize two kinds of solute. One is the good solvent, where the solute remains homogeneously mixed across the solute mole fraction domain. And then there is bad solvent, where the solute like it is inside the binodal, so therefore it phase separates after a certain value of solute mole fraction. With that idea in mind, we wanted to understand how the membrane remodeling processes in good and bad solvent condition. First, we characterized what happens in good solvent condition, and in order to do that, what we did is we in a system of a vesicle, we added solute in the outer exterior compartment, and then we increase the solute concentration progressively. And what we saw is with increasing function of solute concentration, the membrane underwent morphological changes from prolate to dumbbell to budded shapes. And what was very, very interesting is that this process was reversible. So as we increase solute concentration, the membrane forms these budded shapes with closed neck, as you can see here. And as we decrease solute, I mean, if we add more water in this solute concentration, this means we are decreasing the solute concentration. In that case, the budded neck really opens up, and we could arrive again at this prolate kind of shape, which means that the process of membrane morphological change can be reversible as well. In bad solvent condition, we saw a completely different kind of shape changes. So at a very low concentration, we saw that the budded shape forms, but then the membrane neck progressively opens and closes. We termed coined this term like kiss and run, because the neck comes and goes, like the neck is not stable. It fluctuates between open and closed state. And in this plot, you can see how the neck radius is oscillating over time. Yeah, please. Hello. Okay, okay, let me proceed. Yeah, so, and these are the parts where the membrane neck is open, and here it is like the closed neck. And as we increase the solute concentration a little bit more, we saw a complete membrane division, which was really nice. So we saw that on increasing solute concentration, the membrane first formed this closed neck, and then this neck further underwent scission, and that was triggered by a layer of solute molecules that kind of crossed across the membrane neck. And I mean, you can see this in this movie in a nicer manner, that how the solute is triggering the membrane division process. So we plotted the neck radius. We also saw here the neck oscillation between open and closed state. But after 34 microsecond, once the neck closed, it never went back to the open state. Rather, it completely got separated from the mother membrane. And we characterize the contact area, how it, like, once the solute contact area with membrane reaches threshold value, it kind of jumps up to trigger this membrane division process. So we tried to understand this in terms of membrane coverage by solute, and we saw that in good solvent and poor solvent, the solute mole fraction and the solute coverage, they scale in a linear manner. But in good solvent, the slope of the of this line is much softer than that in bad solvent, although the solute mole fraction is in the similar order. And this scales, and this is translated to the spontaneous curvature as well, that we see that the spontaneous curvature scales linearly with solute mole fraction. And in case of bad solvent, the spontaneous curvature is much more increases much more drastically than in case of good solvent. So in this domain, we saw formation of outbuds in good solvent. In this domain for bad solvent, we saw kiss and run. And in this domain, we saw fission. So the outcome of this study is that membrane curvature strongly depends on the quality of the solute. Now, this is the third and the last study we did in this domain, that if we can try to understand, or if we can model membrane into endocytosis of molecular condensates. And the motivation for this study came from this fact that the recent breakthrough in discovery of biomolecular condensates inside cell can potentially mean that there are these condensates which do not have a membrane, like they are called membraneless organelles for a reason. They can potentially interact with the cell membrane as well as the membrane of the organelles, and potentially can be endocytosed, and how the process will then take place. So these membraneless condensates inside cells formed by liquid-liquid phase separation, and this is very similar to aqueous two-phase systems. And this is something we try to model, that how they can reshape membranous compartments. And there are like it's not out of the blue. There are a lot of studies in recent times which actually in vitro studies as well as in vivo, which demonstrated that the biomolecular condensates can drive membrane remodeling process. So here the model that we did was something like this. We had a liquid condensates which formed in the aqueous compartments in the exterior compartment of our simulation box. And we wanted to see when these condensates are representative of aqueous two-phase systems, like that can be ethylene glycol and dextran, or liquid condensates of intrinsically disordered proteins. Here we took the liberty of representing them in both manners because these are coarse grain beads. So how their interaction would be different for different kinds of membranes. And the reference of membranes come from previous studies. We already knew that the membranes behave differently when there is a transbilayer asymmetry. So what we did is we again created these different sets of vesicles as a function of increasing fraction of lipids in the outer leaflet, which would mean the outer leaflet tension will also vary. And we saw very drastic cases of endocytosis. We saw at high outer leaflet tension, complete endocytosis. We saw an intermediate case of leaflet tension where the endocytosis got hindered. And we saw an alternative process of endocytosis as well. So to start with, we started at this, I showed you this plot before, that we plotted leaflet tensions as a function of fraction of lipids in the outer leaflet. And at very high value of tension in the outer, at very low, when the tension is, yes, it's true, like it's like a high value of tension in the outer leaflet and low value of tension in the inner leaflet. So here we saw that these liquid condensates, which are in green, they not only get engulfed by the membrane, but they also get completely endocytosed. So you can see here from the top view that the green point that you see, which is representative of the condensate, disappears inside the vesicle as the membrane gets completely endocytosed. We did it with two sizes. So this is a smaller size. In this case, we saw the complete endocytosis. In case of larger size, we could not achieve complete endocytosis, but still we saw that the droplet got engulfed by the membrane. So in this case, the membrane neck that formed at this interface was circular and axisymmetric. Now there is a case of impeded endocytosis that we saw when the leaflet tension in the outer leaflet is intermediate. So this is the cross-sectional view where the droplet gets engulfed. But if you see the top view, you will see that the green droplet or the green line segment does not disappear. It remains there, and it creates, eventually it gets transformed to a non-axisymmetric shape, and we call this shape a non-axisymmetric tight-lipped membrane neck, because this is like, it reminded us of a lip that is like a long and this thick kind of segment. And then there is this third case, which is the alternative mechanism of endocytosis, where we saw that when the outer leaflet tension is low, in this case also the droplet got engulfed, but in a reciprocal manner, that if you see the final shape, it would be similar of exocytosis, but here actually membrane extended in the form of a tongue on the droplet without forming any permanent pore. It engulfed the droplet, which was very interesting. So what this study actually kind of pointed out is that one can create the process of, or one can, in biomimetic systems, one can achieve the process of endocytosis without incorporating complex protein machineries, which is usually known to be the case, that only complex machineries can trigger such very active cellular processes. So in order to characterize the process, we first calculated membrane segment tension. So as you can intuitively understand, the membrane segment boxed in red here will have very different tension from the membrane segment here. And we wanted to characterize them and find out what is like in terms of numbers. And we found that these values are actually very different. So what we saw is that in case of this segment, which is in contact with the droplet, the tension of the segment increases progressively as a function of lipid in the outer leaflet, whereas in case of the segment here, like which is away from the droplet, the tension decreases progressively. What was very interesting to find out is that we always saw complete endocytosis at this site when these lines did not cross, and after they crossed, we saw impeded endocytosis. So in order to have a successful endocytosis, one has to maintain the membrane tensions, segment tension at these values. What was also important to understand is like how the line tension at this, which gets generated at this two-phase contact line, how that line tension changes. And we calculated the line tension using this equation, which incorporates the segment tensions, the angle that the droplet makes at this contact line, and then also the radius of the contact line here. And if you solve this equation using, we got all these parameters from the simulation, we could actually calculate the line tension, which is here. And the line tension kind of shed light on the process, that how the wetting of the membrane by liquid droplet happens. And what was revealing to us is that below a certain fraction of lipid in the outer leaflet, the line tension is positive, and it's that at that domain we saw the complete endocytosis, whereas at low tension of as the fraction of lipids in the outer leaflet increased, the line tension went downward and gave a negative value, and in that case, we could not achieve complete endocytosis. So, to summarize the mechanical origin of endocytosis of liquid condensates, what is the take-home message here is that when does endocytosis happen and when does endocytosis fail? So what we understand from the study is that in order for the endocytosis to happen, first we need high outer leaflet tension. And secondly, we need positive line tension at the contact line. However, if we cannot achieve that, that is, the leaflet asymmetry is weak in the membrane, leading to low outer leaflet tension and negative line tension at the contact line, we will not achieve endocytosis. So, this study gives us an overview of how one can in synthetic biology, for example, where one tries to minimize the number of components, one can possibly achieve the compartmentalization and endocytosis by keeping in mind the composition of the membrane, and in particular the composition of the lipids in the two leaflets. And we are collaborating with experimentalists where actually we can see that what we saw in simulation, that is, in case of a liquid droplet, which forms in aqueous two-phase systems, the liquid droplet is highly adhesive. They see this kind of engulfment process that we see in simulations. So to summarize the entire study, what we did, we tried to understand, or we tried to develop a new roadmap to membrane remodeling. So this is like the first carried out study in terms of transbilayer asymmetry. It was not characterized how transbilayer asymmetry or inhomogeneous distribution of lipids in the two leaflets can lead to membrane remodeling. That's where we worked at. We tried to give an in a constructive manner. We gave a formula like how one can create inhomogeneous density distribution to transform membrane morphology. What is very striking here is that we also developed methods for new mechanism of membrane division without complex protein machinery, which can generate sufficient constriction force for neck cleavage. And one can do that by taking combination of membrane asymmetry and solutes that rightly get absorbed on the membrane. And to give you an estimate, this is a plot of constriction force in piconewton as a function of curvature difference in membrane. And you can see that the neck fission happens at a very high value of constriction force, and that is only achieved by these complex protein machineries like dynamin, escort, or bacterial protein like FTSZ. And we could achieve such value of constriction force just by tuning these parameters. We also created a minimal model of membrane endocytosis, again without any complex machinery. And the whole study sheds light on the importance of leaflet tension. And that was our goal here, to establish how one can characterize the leaflet tension and then develop biomimetic systems by keeping the leaflet tension inhomogeneity in mind. I will end my talk by giving you a little flavor of what I'm doing currently now with my collaborator from Poland. It's his brainchild, Bartosz Rożki, like who we know each other since a very long time. While I started doing my postdoc at Max Planck, I followed Bartosz's work that preceded in order to develop the studies that I did. And that's how we bonded, and we are still working on understanding a membrane curvature sensing by biomolecular condensates. And the idea stems from the fact, again, I gave you already an idea about it, like how biomolecular condensates can wet and change membrane shapes and membrane curvature. And there are several studies about that. And what we were particularly interested in understanding is how membrane bending or membrane curvature changes by by these proteins called galactin and their condensates. And it is known that galactin machinery is a part of clathrin independent endocytosis. So we wanted to understand if the liquid-liquid phase separation has a role to play there, because these proteins are known to undergo liquid-liquid phase separation. And so here we developed again a coarse grain model using dissipative particle dynamics, where we had two kinds of lipids. And we also modeled a polymer, which is kind of representative of galactin-3 because it has a very long tail and heads that particularly recognize these lipid B here, the head groups of the lipid B. And first we characterize the phase separation of the polymer in solution, and then we carried out the study further to understand how these phase separation of these galactin condensates can execute membrane curvature sensing. So we saw that the lipid B, in absence of these polymers, they don't phase separate, but they separate on the membrane. But when there is this in presence of the polymer, the lipid B undergoes clear phase separation. And we are extending this work further and doing some very interesting modification to the model. Yeah, so with that, I would like to conclude my talk. I would like to thank my supervisor, Professor Reinhard Lipowski, and my experience there was life-changing. And all the studies that I did was done at Max Planck Institute of Colloids and Interfaces. There, I also collaborated with, and still collaborating with, Rumia Nadimova, who is the biophysicist there for the experiments. And the ongoing collaborations are with Bartosz and Amir. And with that, I would like to thank you for your attention and would welcome any questions that you might have.
Thank you, Rikhia. So we have some lost. Okay, so now we are open for discussion. I would suggest everyone who is interested in asking just to raise a hand and unmute. Use the mute button. Okay. Okay, so I will start asking some questions. Maybe it's a question I had in the past to Bartosz, but it's about the coarse grain model of lipids using the DPT. So, I mean, as you explained, so it has a lot of advantages to develop a coarse grain model, as you presented through all these examples. So the parametrization of the lipids must be a little bit based on some all atom or experimental data. So how did you model the parameters for your lipids?
So for the lipid model was kind of like done or built keeping in mind the lipids which has like POPC, or like the generic lipids that one finds in cell membranes, or like one often uses while creating these biomimetic systems. And so this model is kind of well known for these lipids. And what is done there is like a lot of biophysical studies in order to confirm that the lipid diffusion is in correct order. And also, like, for example, like how the interactions are different for different kinds of lipids. So, but I mean, I would not encourage you to read too much into it, because this is a very, very coarse grain model. So you can practically change this conservative force to create a lipid of the kind that you might want to. So you can basically add more beads to create, like that's what I did, like we did with Bartosz. So what Bartosz wanted to create was this large head lipids. So he added lipid beads to the existing model. So I mean, the physical values are not really like transferable to experimental values. But I mean, what really encourages us is that even though this is very coarse grained, the phenomena that we can observe or the stress profile, or I mean, we can really tally the observations or to for example, it's very well known from curvature elasticity models that membranes can form certain shapes at certain curvature value. And that's something we could reach at while when we uh, underwent this study. So that in a reverse way, that confirms that the lipids we have are kind of generic, represents the lipids that are biologically available. Sorry, I can't hear you.
I said that I might have another question, but it can go after some. If somebody else doesn't have a question, then I will ask. Please, guys. Anybody in the audience? Oh yeah, there is one there.
Yeah.
Fernando.
Very interesting talk. Thank you very much. I got a question more about possible applications. I don't know if this could be applied for pharmacological uses, for example, to model vesicle-like formulations to deliver drugs in the body.
That's a very good question, actually. I mean, so in that case, this is actually my goal moving forward. I want to, because now I'm working on drug discovery. This is something I really want to work on, and I have plans. So there, actually, I would rather not take this model because it does not have a lot of chemical features. So but what one can expect is that you will find same observations even when you have a more detailed system. Like in case of martini, you have chemical features. So if you can create a vesicle in martini, and then have, because in martini the standard, like you might know that they are already very well equipped to model the coarse grained drug molecules. So I would say that's a very good starting point for studying the drug delivery process, having created a membrane in martini, and then see how the interaction of a drug molecule with the membrane occurs. And I'm sure that will be very, very promising study, because this is not something that is done. You can go to larger time and space. You can see the endocytosis of a drug, like how that permeates a membrane. So that would be a very interesting study to execute.
Okay, thank you.
Maybe
you're
you are
mute again.
Yes.
And I don't know what is happening, but okay. Yeah, there is one. Somebody else. Yeah. Hank could just ask the question.
Hi, now I can activate the microphone. Thanks for the presentation. My question is regarding to membrane curvature. How did you calculate the curvature of your membrane?
So in this case, what we as I was explaining, that we calculate the membrane tension, that we calculate from the stress profile. And the first moment of the stress profile is related to the spontaneous curvature of the membrane. If you like, I can send out the details to you about how the equation evolves. So this equation has the bending rigidity and the spontaneous curvature, and they are if you know the bending rigidity of the membrane, there are ways to calculate that from simulation. And you can also calculate the stress profile. So if you calculate the first moment and incorporate the value of bending rigidity there, you will get the membrane spontaneous curvature. So you
Nice.
also need to have a mean curvature there. And the mean curvature for a vesicle is simple. It's an inverse of the radius. So you will get the spontaneous curvature directly from that.
Okay. Thanks so much. And I'm so interested in detailed equation. I can text you later then. Would be very nice to see your way. How did you did it? Thanks so much.
Thank you
Then
so much.
I will keep in contact, and for the equation, I want to see the detail because I was trying to calculate from a different kind of example. It's not working there. That's why it would be very nice to see your equations. Thanks so much.
Absolutely. I will be very happy. I will send you over the equations. It is fairly simple to calculate from simulation, so it shouldn't be hard.
Perfect.
Thank you.
Okay, so we keep the discussion. Any additional question? I have one, and this is about the fluctuations. So in this model, so because this is molecular dynamics, and what you parameterize, as you said at the beginning, is some diffusion, some kind of, I mean, the conservative part of the DPD is kind of the part which distinguishes the interactions between different type of lipids, yeah? And
Yeah.
so finally, what you get out from MD simulations are just fluctuations. So are those fluctuations comparable, at least in terms of the all atom simulations? I mean, is the system not only diffusing, but also having the right fluctuations? Because in the end, so whatever bends or gets this remodel the shape, it has some specific fluctuations. Are those comparable to all atom simulations? If ever one can do such a simulation, so would you expect the same fluctuations?
I would say so. So this is something that's a bit tricky, like because this scale, like we have a diameter which is like 50 nanometer, and then like the time scale is of the order of 100 microsecond. So this is something that's, at least for this kind of studies, like this was our reason to choose something very coarse, because you cannot go to this scale using even Martini. It's not easy, like because of the limitation in computational resources, like so. And we wanted to see at the same time the shape transformation, so we needed to have the vesicles. So to answer your question, I would say it is, I would like to believe it is comparable. I mean, I'm not saying that you can match exactly at what time scale and what length scale this is going to occur. But definitely, this is like also molecular dynamics using the, I mean, we need to conserve the momentum here, for example. So it is definitely going to be comparable to other kinds of simulations.
Okay. Is there any additional question? Oh yeah, another one. Okay, Han, go ahead.
Please.
No, maybe there was another question. Okay. Yes, there is a question. Why it's not?
Now the microphone, because I can't unmute it.
Okay.
My other question is related to membrane type. In your presentation, your work is based on PubC and PubE lipid type. Have you ever tried different kind of lipid like PIM or PIP? Because some bacteria, they are like, especially membrane flexibility, totally different than PubC, especially for non-symmetric membranes.
So this is what I was trying to convey, that this is a very coarse grained membrane. And I mean, there is no charge involved. So you cannot read, I mean, if you want to go to a detailed chemical nature, it's better to use a different kind of force field, because this is the limitation of this force field, that you cannot retain a lot of chemical feature other than the interaction, like how repulsive or attractive are the interactions, or how large are the, for example, the lipid heads, or how long are the tails. You can build up on that. But what you mentioned is, yes, I mean, I completely agree that different lipids have different chemical natures. So here, the only nature that you can really tune or work around is like the hydrophobicity and hydrophilicity. But if you want to add more to that, it is better, like it is definitely you need something which a model which has more chemical features.
And also your collaborator, they are experimenting in the wet lab study with the other type of membrane, right? Because
So for this particular study, they had a pure POPC GUV. GUV means giant unilamellar vesicle. So it's an in vitro biophysical study. So it's very simple membrane.
Okay.
But what you asked, like it would be interesting to actually see what happens when the membrane has more chemical features, which we don't have yet.
Okay. Thanks so much.
Thank you.
Anybody else? I don't see any more questions. It's getting here a little bit late.
Yes,
For
I
you,
am.
it's a little bit earlier. That's good. You have the full day.
Yeah, that's
Okay, if there is no more questions, I guess I can close the session. And I really say thank you, Rikhia. It was a very interesting talk, and we hope to stay in touch also. Okay.
Yes, definitely. And
And
thank
you are
you.
yes, and you're welcome to be again here, maybe with some experimental, or maybe Bartosz also. I
Yes,
will
that would
contact
be
him.
very nice. Bartosz is doing a lot of work on this, so
Mhm.
it would be nice for
And he
him.
is very busy also, I guess.
Yes. Yeah.
Anyway, thank you once again. Okay.
Thank
And have a nice day.
Bye. Have
See
a
you
nice day,
soon.
everyone. Thank you. Bye.
Bye bye.